Inside of the prokaryotic cell, the cytoplasm. The cytoplasm is the substance inside the plasma membrane. It is 80% water since we don't have membrane-bound organelles. There is less water in the eukaryotic cell due to membrane-bound organelles. It contains all internal structures. Instead of a nucleus, bacteria have a nuclear area or nucleoid. The nucleoid contains the DNA. The DNA is not membrane-bound and there are no histones. The DNA is usually arranged in one large circular chromosome. So it's a circular single bacterial chromosome that has 4,300 genes. It is weakly bound to proteins in the plasma membrane that help it to replicate and separate during cell division. It is about 20% of the cell. Some bacteria also contain small circular molecules of DNA called plasmids. Genetic information in plasmids supplements information in the chromosome. The cell can contain one or more of these extrachromal DNA, which are about 5 to 100 genes. It is often transferred to other prokaryotes by conjugation. It contains non-essential genes such as antibiotic resistance. So when we say non-essential, the cell does not require that it's antibiotic. It doesn't require to be antibiotic resistance. It doesn't need it to survive. Ribosomes is the site of protein synthesis. It consists of RNA and protein. For prokaryotic cells, it is a 70S ribosome. The 70S ribosome contains a large subunit, 50S, and a small subunit, 30S. So large subunit and a small subunit. And that is where we get 70S. So you're probably sitting here like, okay, Miss McFarland, you in the book cannot add. Well, the relative size of ribosomes and their subunits can be determined by measuring their sedimentation rates, the rates at which they move towards the bottom of a tube when the tube is rapidly spun in an instrument called a centrifuge. As they're spinning, the sedimentation rates are expressed in the terms of Spurberg units. So the S represents Spurberg units. So they got 30S and then 50S, but altogether the sedimentation rate is 70S. Bacteria can have within their cytoplasm a variety of small bodies collectively referred to as inclusion bodies. Some are called granules, others are called vesicles. Inclusion bodies are storage granules that hold the cell's reserve supplies of nutrients. Inclusion bodies vary from species to species. Metachromatic granules are phosphate reserves, so they store phosphate. Polysaccharide granules are energy reserves. Lipid inclusions are also energy reserves. Gas vacuoles are vacuoles filled with air for buoyancy. Some photosynthetic aquatic bacteria have these gas-filled regions surrounded by a protein membrane that allows the bacterium to float at the water level that provides best conditions for photosynthesis. So if they're at the bottom of a lake and then it's a lot of light, they can release these gas vacuoles to be able to rise to the top of the water. Okay, the polysaccharide granules, if we go back to these, they're usually consisting of starch or glycogen. So that's what we mean by energy reserves. So lipid would be stored as energy reserves. So again, these could have starch or glycogen. Magnetosomes are ion-containing structures, and they are inclusions of iron oxide. So you have iron oxide in there, which will destroy hydrogen peroxide. Endospores are resting cells that are highly durable. They are resting structures that are non-growing, that could survive up to 100 that can survive up to 100 years and are highly resistant to heat, desiccation, which is dehydration or drying, acids, bases, certain disinfectants, and even radiation. So nothing will kill them unless you're using an autoclave. It is a survival mechanism for some species. So for the genera Bacillus and Clostridium. So we already learned Bacillus anthracis that causes anthrax, Clostridium botulism, botulism, Clostridium tetani, tetanus. Conditions that might lead to sporulation. Sporulation is endospore formation. Is if they're in a too hot environment, too cold, too dry, exposure to chemicals, and a lack of food. So if they have low nutrient availability. So if there's no food available, they will form these endospores. Usually when an endospore forming species stops growing, it starts forming endospores. Germination is when a spore returns to its vegetative state. Once favorable conditions occur, such as an increase in temperature, water increase, or nutrient increase, it develops into a vegetative cell. Vegetative cells are cells that grow and reproduce. But vegetative cells lack the endospores' resistant properties, so it no longer is resistant to heat or chemicals. It's very easy to kill at this time. Here ends Chapter 2.